What if the DNA inside certain people alive today is the closest living proof we have that the Bible's most powerful tribe actually existed? In 1997, a single study traced one male lineage back 3,000 years to the ancient tribe of Judah. 5 genetic secrets now point to the same conclusion, and the first begins with a signature in living blood that should not still exist. The hereditary priesthood, or Kohanim, presents an unusually clean testing ground for genetics because the priestly title passes strictly from father to son. In 1997, researcher Karl Skorecki published a study in the journal Nature analyzing the Y chromosomes of these self-identified priests. He discovered a recurring genetic pattern, now known as the Cohen modal haplotype. While only 5% of the general non-Jewish population carries this specific marker, it was present in about half of all the Kohanim tested. Using standard mutation rates, scientists estimated that the common ancestor of this lineage lived roughly 3,000 years ago. This timeline converges directly with the early Iron Age, the period when the ancient priesthood was first established in the southern Levant. The geographic distribution of this marker points to deep historical roots. Despite being separated by thousands of miles and more than 500 years of complete geographic isolation, both Ashkenazi and Sephardic Kohanim carry the exact same paternal signature. It is one of the most striking examples of genetic identity preserved across millennia. Recent ancient DNA analyses from Iron Age southern Levant sites, dating between 1200 BCE and 600 BCE, reveal a high concentration of Y-chromosome haplogroups J1 and J2, the same parent lineages containing this priestly signature. Rather than validating theological beliefs, these markers provide concrete biological evidence of physical, generational continuity, linking living populations directly to the soil and the historical people of ancient Judah. In southern Africa, the Lemba people of Zimbabwe and South Africa have long maintained oral histories of Jewish ancestry, practicing male circumcision, avoiding pork, and keeping a Saturday Sabbath. These cultural markers, distinct from surrounding Bantu-speaking groups, prompted British scholar Tudor Parfitt of the University of Oxford to investigate their genetic makeup in 1999. Testing focused on the Buba clan, the Lemba's traditional priestly class. Parfitt discovered that 53% of the Buba males carried the exact same Y chromosome signature found in the Middle Eastern Kohanim. This remarkable frequency aligned the African clan directly with the ancient priestly lineage. Further north, another isolated group preserved an ancient Israelite identity. The Beta Israel of Ethiopia, airlifted to Israel during Operations Moses and Solomon between 1984 and 1991, hold an oral tradition linking their lineage to the biblical tribe of Dan and King Solomon's court. While autosomal DNA shows heavy local African ancestry, a 1999 Tel Aviv University study identified a measurable frequency of the Near Eastern J1 haplogroup among the Beta Israel. This specific paternal marker distinguishes them from neighboring East African populations. While the genetics reveal a complex history of regional integration, the presence of these ancestral markers does not contradict their historical claim. Instead, it expands the geographic footprint of Levantine genetic traces deep into Africa. The genetic signature of the ancient Levant reaches far wider than isolated families. Today, Jewish communities are dispersed across more than 100 countries. Yet the paternal lineages of Ashkenazi, Sephardic, and Mizrahi men remain unusually clustered. At the population level, the combined rate of Y chromosome haplogroups, J1 and J2, exceeds 50% across these geographically separated groups. Scholars at the Hebrew University of Jerusalem have described this persistent clustering as almost unprecedented for dispersed ethnic populations. A 2010 genomic study published in the journal Nature by geneticist Harry Ostrer examined this genetic closeness across the diaspora. The data revealed that Jewish men from Morocco, Germany, Iran, and Iraq cluster far more closely with one another than with the non-Jewish populations living in their respective host nations. This high degree of genetic proximity reflects a historic bottleneck, where a population is drastically reduced to a small number of ancestors. The timeline for this contraction aligns with the historical trauma of ancient Judah. In 586 BCE, Babylonian forces destroyed Jerusalem and deported much of its population. In 538 BCE, Cyrus the Great issued a decree allowing a small remnant to return and rebuild. This founding population established a tight pattern of endogamy, locking the J1 and J2 paternal markers into the gene pool before the Roman-era dispersals. While this clustering functions as a broad population signal rather than a simple label of personal identity, it prompts a final question. Paternal lineages show a clear geographic origin, but does the maternal line tell a similar story? Mitochondrial DNA, passed exclusively from mother to child, offers an independent genetic archive that bypasses the male line entirely. In 2006, a study published in the American Journal of Human Genetics analyzed the mitochondrial genomes of more than 11,000 individuals. The researchers discovered that despite centuries of geographical separation, the maternal lineages of Jewish women across diverse diaspora communities cluster tightly with ancient populations of the Near East, specifically the historical Levant region, encompassing modern Israel, Jordan, and Lebanon. The data suggested that even after more than 1,000 years in Eastern Europe, European maternal admixture remained low, at less than 20%. However, in 2013, a study published in Nature Communications by Martin Richards refined this maternal portrait. Richards identified 4 specific founding mothers who account for roughly 40% of all modern Ashkenazi mitochondrial lineages. While the geographic origin of these maternal ancestors remains a subject of scientific debate, the demographic footprint of their legacy is clear. This maternal concentration represents a classic genetic founder effect, a phenomenon that also left a distinct signature in the community's medical history. Rare recessive mutations carried by these few ancestral mothers became increasingly common over generations of endogamy. Today, genetic disorders like Tay-Sachs, Gaucher disease, and familial dysautonomia occur in these populations at rates nearly 100 times the global average. Remarkably, identical founder mutations appear in Jewish communities historically isolated in Poland, Morocco, Ethiopia, and India, despite centuries without documented contact. These shared genetic markers point to a common maternal origin that survived geographic dispersion. But to understand the complete genetic architecture of this population, researchers had to look beyond single ancestral lines to the whole genome test. In 2010, geneticist Doron Bahar and an international team of researchers published a study in the journal Nature that shifted focus from isolated paternal and maternal lines to analyze the entire genome. By scanning hundreds of thousands of single nucleotide polymorphisms, known as SNPs, across more than 1,800 individuals from 106 different populations, the study tested the genetic architecture of diaspora communities. The analysis revealed that genome-wide data can identify Jewish ancestry with over 99% accuracy. When mapped mathematically, Ashkenazi Jews did not cluster with their historical Polish or German neighbors. Instead, they grouped closely with Sephardic and Mizrahi Jewish populations from Morocco and Iraq. This persistent proximity across vast geographic distances points to a long history of deliberate endogamy, a practice of marrying within the community emphasized in the biblical texts of Deuteronomy and Ezra. While other ancient Near Eastern populations gradually assimilated and vanished, the descendants of Judah maintained a biological and cultural coherence through centuries of dispersion, a phenomenon that lacks a clear parallel among other ancient groups. Yet, geneticists maintain a strict methodological boundary. Autosomal clustering is probabilistic. It maps shared history and population continuity with high statistical confidence, while remaining silent on personal religious status or theological proof. What it does provide is a concrete biological archive. Across maternal lines, paternal signatures, and now the entire genome, the genetic data converges on a single historical reality. Today, molecular biology achieves what archaeology alone could not. For Judah, the ancient text and the genetic record finally speak the same language. This enduring biological signature proves that history is not merely written, it is carried. Share your thoughts in the comments below.